Hello, welcome to video 33 in my lecture Advanced Mathematic Modeling. So we are discussing random walks, and in the last video I showed you this example of a two-dimensional random walk, this Matterhorn-de model, where we looked for the displacement in x direction, and we saw that the increments in x direction are long-range correlated, so the outer covariance function decayed as a power law of the time distance. So here's the power law, and as a consequence we have seen that the mean square displacement is increasing with time as a power law with an exponent which is not 1 over 2, not 1, like if you have here x the second moment, so it's not a 1, but it's 3 over 2, it's larger. And so we had the case of a walk which is not in the class of random walks. So it's not a normal, not in the class of diffusion, it's in a different, not in this universality class. And such processes we call super-diffusion. So the super-diffusion are random walks where the mean square displacement increases with an exponent as a power law with an exponent that is larger than 1. And the reason for the super-diffusion is that the increments are correlated as a power law. And now I want to show you another example of this, and sometimes it's not so obvious when you look at a system that this corresponds to a random walk. And I will show you a famous example, that's the DNA walk, which again is leads to a super-diffusive random walk because increments are long-range correlated. So, okay, so of course, what's DNA, and so we're living in the time where we have a lot of gene sequences. So let's say you have a gene sequence. Yeah, and now we can sequence the genes of the genomes of many organism types and species. And so basically, such a sequence is just a sequence of nucleic acids. So we have the sequence of nucleic acids, and these come as pairs. So we have this base pairs, and so what acids do we have? So we have adenine, thymine, guanine, and cytosine, and they come in pairs, and typically on our base pairs, and then on our always adenine, I always abbreviate them with the capital letter. And then we have pairs of AT, they come always together, or G and C come together. And then if you have now a chromosome, and along the chromosome you have the sequence of base pairs, and so then you have something like this. So you could have something like C, A, T, T, C, C, G, A, T, G, G, whatever, and then you have your sequence. And then of course on the you have always the corresponding matching pair here below. And yeah, and so you sequence this along a chromosome of an animal, maybe a mouse or some plant. And so now the question is, what's the structure in the sequence of pairs. And a lot of microbiology currently is sequencing a lot of organisms and try to figure out what's the properties of these genomes and what can be learned from them. And then some guys came to the idea, can we interpret such a sequence as a random walk? And yes, why not? We can say that's basically a stochastic process. So we have our time is then the order along the chromosome, along the sequence. And then we have at every time instance, or every, let's call it time, every time instance we have one of four possibilities. But since if you want to have just a random walk which goes just left and right, so we can do the following. We just look for which kind of pair do we have. Do we have an AT or a GC pair? And don't care if you have no A or T here. So we interpret an AT pair as +1 and the CG pair as -1, and these are the increments then of our random walk. And now we can just sum these increments up, and so we will obtain a random walk. So let's see what this will be. So here we have our index I. Here we have our x, so the position of the random walk. That's the sum of the first I increments. And let's say now we have here a sequence. So we have here C, A, T, T, C, T, G, T, T, C, T, whatever. And then now with C we go a -1, so we start with 0 and we go one step down. Now we have an A, so we go one step up. We have another A, we go another step up. We have a T, we go a step up. We have another T, we go a step up. Now we have a C, we go a step down. We have a T, go a step up. G, go down. T, go up. T, go up. C, down. T, up. And so we obtain a random walk. And this construction we can of course do. It's of course it's not clear what should be the biological interpretation of this. What should be the sum of n increments which we get like this. So that's x directly probably has no biological meaning. But that's not the point here. We don't want to try to figure out what does it mean to have a certain x value in this position. That's not the point. The point is that we obtain here a random walk and try to find out, does this random walk have certain properties. And as always with a random walk, if we do here 10 or 20 steps, we don't see so much. But let's say we do much more. And for example, you can now do or maybe let's write down here first the question here. So x itself let's say has no biological meaning. At least it doesn't have a biological meaning that comes directly to my mind. But maybe the properties of the associated random walk. And that would be a question. So does this random walk that we obtain here when we analyze this with our normal statistical analysis, does this have properties which can mean something biologically? Okay, so now how does it look if we take a larger genome? So let's take a sequence of 20,000 base pairs. So here we have our index I, maybe you have here 20,000, here 10,000, 5,000 base pairs, and that's nowadays just a piece of cake to do this. To have this genomic information, you find them also in big databases, even much longer nowadays. And now you calculate the random walk, compute the random walk, and maybe you find a profile which looks like this. And that's the position of the walker. Now we ask, what's the properties of such a random walk? Yeah? Does this random walk show diffusive behavior or not? And so what's our typical way to analyze this? So we make a double logarithmic plot where we take here the log, and we take log 10 of our index I, and here we plot log 10 of our square root of the second moment. So we plot the mean square displacement. And if one is doing this, and so we go here, if we have log 10 here, so how many orders of magnitude do we go? So we start here from 0. This would mean log 10 of 0 means we have just one time step, and then we go here to 1. That would be 10 time steps. 200. 3 would be 1,000. 4, 10,000, and we go to 20,000. So that's the range in log 10. So we can analyze the mean square displacement over 4 orders of magnitude, because we have increments, time distances of 1 step until something like 20,000. And so now one can, now the problem is we have now only one profile, so we cannot average here over many different profiles, or if maybe you have the same species sequence still a lot of time, then you can average over many sequences. Or we do the trick that we had seen before. We take different starting points along the sequence and average over different small intervals that we randomly choose out of the sequence. And then we now plot here the mean square displacement over the time distance, and take time distances nicely equally spaced, so that it gives a nice fit in the log-log plot, maybe something like this. And we see that one can put here a straight line through this data over some range, some scaling regime, and then for very large time differences the scaling is lost here. And so we see here a scaling, a straight line behavior. We have here a scaling regime. And now we can, for example, make a straight line fit here, and we find an exponent alpha which is about 0.6 to 0.7 here. So we find that the second moment square root as a function of time goes proportionally time to a power, an exponent alpha, and this alpha is larger than 1 over 2. So we have a super-diffusion. So a result which is very similar to this what we saw here in the Matterhorn-de-Marcilly model. Just look for the difference here. We analyzed just the second moment, and then we have an exponent which is larger than 1. And if we here, just for variation, I plotted the square root, and then the critical exponent will be larger than 1/2. Yeah, so that's just arbitrary if you want to analyze the square root or the second moment. But in both cases we find a super-diffusion. But you see, doesn't go over the whole range of the whole measurement, and this was taken from the original publication. So and it's for me not clear if the saturation now is because if you average take intervals here which are larger than maybe something like the half of the time series, you have not many points to average out. So is this here already a sign that we didn't have enough long enough sequence, or is really something happening after maybe something like distances of about 1,000 base pairs? So that's not so clear, but interestingly we find this super diffusive behavior. And now the question is, we can now come back to the biology. So but first, so you see, even in systems where you wouldn't directly think about this, there's always the chance that you can interpret some data, for example just a genomic sequence, as a random walk, or you interpret the data as increments and then construct the random walk, and then you can try to analyze this random walk. So now what does the biology mean? So we have super diffusion here. So so how possibly can we have such super diffusion? At this point maybe it's we should remember again, so how when we calculated the mean square displacement. So what determines the mean square displacement? We calculated this. So we have n times here the variance of the increments plus this correlation sum. Now the second moment of our increments is definitely 1, because we constructed it like this. So so we would expect, so this is 1, so we would expect that the mean square displacement goes proportional to time, so we would expect diffusion. And so the only thing what can happen is that we have here a correlation, a covariance function, which is increasing, which is not decaying, but is increasing or is decaying very slowly with time, so probably as a power law, just as we had it in this Matterhorn-de-Marcilly model. Yeah? So that's the only explanation that we have. So the biological meaning is that the increments are correlated over long distance. So explanation: that the base pairs, so in our language the increments of the walk, are long range correlated. This means there in such a sequence in the genome there is a memory, and so if we have a certain base pair at a certain position, then the chance that neighboring base pairs have a certain value depends on the base pair in the neighborhood. Yeah, so there is a memory here, there's a correlation, and this correlation doesn't decay exponentially, decay very fast, I mean, we had this with our Taylor approximation. We have seen if we have a short memory, so a short-term correlation, then we would eventually find here normal diffusive behavior. But since we don't have diffusive behavior because we have here super-diffusion, this means that this autocovariance function is long-range correlated. So this means the presence, if here is a CG or an AT, yeah, has an influence on base pairs which are 1,000 steps further away. Yeah. And now you can start to think, what does this mean if we have here long-range correlation in the sequences? How can this possibly come from? And then we come to the point where we might learn some biology. And in the exercises you will analyze a simple model for such a DNA. So we call this DNA model, a model for the generation of such a sequence. And so how does such a sequence come? I mean, the sequence is generated in evolution. You start from a common ancestor. In our model we have an ancestor, and so this is just the sequence, and so we could say the sequence, we just start, okay, we model just the increments plus minus 1. And let's say we have an ancestor which has just one gene, and this gene is plus 1. Yeah. Of course, I mean, you don't have a living organism with just one nucleic acid, but we could have started with something different, but for the model we just start with one of the most simple sequence. So now what's happening? Now we allow offspring of this ancestor, we allow it to multiply, and every offspring will have a mutation. And we have mutations. And now what kind of mutations can we construct? So like we can say then, so we can either make a random flip. Let's say we have a sequence here. Here's such a sequence, and then we take a random gene and just flip it. So we take a random, let's say a random nucleic acid, which is in our model just plus or minus 1, and we just flip it randomly. So we have random flips. So the position in our sequence is just multiplied by minus 1. Then we have random deletions. So this means just one random taken nucleic acid is just deleted from the sequence. Oh, we should go a bit up. And then, but if you would have only this, and maybe if you make random insertions, so like let's say we have here sequence EI minus 1, EI plus 1, EI plus 2. Yeah, so that's your sequence. So means so like we take one random of these increments and either flip the sign, we just delete it, or maybe we just insert at a certain position here a random increment. Yeah. If you would do these three processes, always random do it, and then always our sequence is changed. But if you would do it and then calculate the walk, you would have a normal diffusive walk. Yeah. And the trick to find the super diffusive behavior is that we have a duplication. Random duplication. This means, for example, if we have here a certain increment here, and we take the same increment and put it 2 times at this position here. So we duplicate just a random sequence. Now this random duplication makes a short-term correlation. Yeah, because suddenly we have a pair of nucleic acids which are the same. But how can this random duplication, how can this become long-range correlated? And then the trick is, let's say we start with a 1. That's our ancestor. Now we maybe by chance have a duplication, and then we go to 1, 1. So that was the first sequence, and then we have this. So we introduced some correlation. And now we go further, and then we still have this 1 here. We make a lot of insertions, and then later this one, after many insertions, shifted here very far to the right. And so this, because if we have here by the duplications we introduced some short-term correlation here, and then if we now insert other genes here in between, so this short-term correlations due to insertions becomes suddenly a correlation over longer time distances. But during these insertions we always put here new random stuff, and this also with a certain chance have duplications, and so there is again some correlations appearing, and these correlations again after further insertions are again longer range correlated. So by doing these duplications and then always inserting stuff, in the end we end up with a sequence where we have correlations of all orders of lengths, and this leads then to the power law correlations. And okay, you will analyze this model in the exercises, and because you can simply create such a process and then see that you find the super diffusive behavior. And now we can take such models as a test bed to see what kind of mutations you can have in the evolution of a genomic sequence which is consistent with the long-term behavior, the super diffusive behavior. Yeah, and then you see suddenly how such statistical analysis can help to learn something about a certain system. In this sense, what we can learn about the evolution of genomic sequences. Okay. So maybe it was a bit detailed, this example, but I just wanted to show you how suddenly the random walks can emerge suddenly into systems, and the importance then, if we find something which is not normally normal diffusion, that this leads then to certain restrictions in the processes which are take place. And here we see that this duplication events, you can have either duplication of a single sequence. In reality you have duplications of larger parts of sequences, that such duplications play probably a large role in order to explain such long-range correlations. Okay, for now, thank you.